The next topic of DNA, we are going to learn about structure of DNA or double helix structure of DNA. J.D. Watson and F.H.C. Crick first proposed the structural model of DNA in 1953. They got the Nobel Prize for their work in 1962. According to Watson and Crick model of DNA, the DNA contains two polynucleotide strands coiled together in helical manner. So we learned what is polynucleotide strand or polynucleotide chain in the previous part. So here what they told, the DNA contains two polynucleotide strands. They coil together in helical manner. Hence, the name Watson and Crick double helix structure of DNA is given. You can see here the picture of Watson, James, the full name it is James D.V. Watson, born April 6, 1928. He got the Nobel Prize at the age of 34 in 1962. This is a picture of Crick, Francis Harry Compton Crick. He lived 8th June 1916 to 28th July 2004. The structure of DNA is as follows. Let us see what is the structure of DNA. The DNA is a double-stranded polynucleotide molecule. So it is made up of two strand or two chain of polynucleotide. It is a double-stranded polynucleotide molecule. Sugar and phosphate forms the backbone. So we learned it is made up of sugar, phosphate and a nitrogen base. So here sugar and phosphate forms the backbone. The bases like cytosine, guanine, adenine, thymine, everything is arranged, projected into inside. The two strands are coiled each other and arranged anti-parallelly. That is, if one strand has fifth to third and the other has third to fifth in direction. So we, how is bonding? We learned in the previous section. So how it is arranged? One strand has fifth to third and the other has third to fifth in direction. The two strands of DNA have the common diameter of 20 degree angstrom. So if you are taking two strands of DNA, they have the common diameter of 20 degree angstrom. Adenine of one strand pairs with thymine of another strand. So there are two strands. Always adenine pairs with thymine, adenine a capital A and capital T. That is a pair. Strand by two hydrogen bonds, vice versa. Guanine of one strand pairs with cytosine of another strand. Guanine capital G, cytosine capital C. So that these are the pairs, cytosine and guanine, of another strand by three hydrogen bonds. So adenine thymine paired by two hydrogen bonds. Guanine cytosine is paired by three hydrogen bond. Because of complementary base pairing arrangement, if one strand of polynucleotide sequence is known, another can be deduced. So example, they given one fifth strand, they given A, G, C, T, 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 A, like that is going. So you can see here always adenine. So if you are getting one strand, you can write easily the second strand. So adenine here means opposite will be thymine. Guanine means cytosine. Again thymine means adenine. Third, here cytosine, guanine, thymine, adenine, thymine, adenine, thymine, adenine, like that it is going. It means always these are the pairs. So we are getting this strand, you can write the another second, this polynucleotide strand. The complementary strand twisted each other at the distance of 34 degree angstrom. Each twist of DNA contains 10 base pairs. How many base pairs? 10 base pairs. Each twist of DNA it contains 10 base pair. Which are the base pairs? Cytosine, guanine, adenine, thymine. These are the base pairs. The distance between these two, excuse me, the distance between these two base pairs are 3.4 degree angstrom. The distance between two base pairs, 3.4 degree angstrom. That is the distance. So here you can see, so these are the backbone we told, sugar and phosphate, that will become the backbone. This is the third bond. Here this is the fifth position. So here it will be fifth, here this will be third. So nitrogenous bases, it is arranged inside cytosine, guanine, thymine, adenine. These are the pairs. Guanine, cytosine, again thymine, adenine, like this. Then this is known as major groove. This is the minor groove, small one, minor groove. This is major groove. Then these are the twists. Here the distance will be 3.4 nanometer. But the distance between two base pairs, that is 0.34 nanometer. The distance between two endings, one is third bonding, one is fifth one. So the distance between these two, it is 2 nanometer. The same thing also you can see here. So this is the phosphorus, minor groove, major groove. These are the base pairs in the center. Then here oxygen, then hydrogen, then backbone, that is sugar and phosphate. So sugar and phosphate, that is the backbone. The center you can see nitrogen bases are arranged. Then what is meant by central dogma? Central dogma, this is proposed by Francis Crick. Francis Crick proposed the central dogma of molecular biology, which states that genetic information flows from DNA to mRNA. mRNA means messenger RNA, that is known as transcription. And then from mRNA to protein, that is known as translation. Always this is unidirectionally, but except you can see in some viruses, you can see it is bidirectionally. In some viruses, the process is known as reverse transcription. So how the movement of one-way flow of genetic information from DNA to protein is called central dogma. That is, first from DNA it goes to RNA or mRNA, messenger RNA, that is known as transcription. From mRNA it will go to protein. Okay, this way of flow always it will happen. But in some case of viruses, we can see the reverse cases also we can see. So that is known as reverse transcription. Clear? The next one is packaging of DNA in prokaryotes. How the DNA is arranged in or packed in prokaryote. Prokaryote means nucleus without nuclear membrane. Clear? Nucleus without nuclear membrane, that is prokaryotes. Prokaryotes do not have definite nucleus. The DNA is not scattered throughout the cell. So there is no nucleus. Usually we learn inside the nucleus we can see the chromosomes. In the chromosome we can see the DNA. But here they don't have definite nucleus. So DNA is not scattered throughout the cell. Then where we can see it is held together with some proteins in a region is called nucleoid. So the DNA is tied together or held together with some proteins in a region is called nucleoid. The DNA nucleoid is organized in large loops held by proteins. Clear? So DNA is connected to or tied with proteins in a region is called nucleoid. The next one is how the DNA is packing of DNA in eukaryotes, or how it is packed in eukaryote. Eukaryote means true nucleus. In eukaryotes, DNA is stabilized with positively charged. DNA actually it has negative charge. So it is joined with positively charged basin proteins. They are called histones. Okay, you can see here this purple color, this is DNA, it is negatively charged. These histones, these are the histones, they are positively charged. So together it is called nucleosome. So in eukaryotes, DNA is stabilized with positively charged basic proteins called histones. Histones are positively charged due to rich in basic amino acids like lysines and arginines. So DNA negative, histones positive. Why it is getting positive charge? Due to rich in basic amino acids like lysines and arginines. Histones are organized to form a unit of 8 molecules called histone octamer. You can see here 1, 2, 3, 4, 5, 6, 7, 8 molecules together arranged. These are the H1 histone. So these are always 8 molecules combined together. They are called histone octamer. Octa means 8. So this is called histone octamer. So histones are organized to form a unit of 8 molecules called histone octamer. Clear? So this histone and DNA, these are DNA. Together it is called nucleosome. Now, negatively charged DNA wrapped around positively charged histone octamer to form a structure called nucleosome. Already I told, negatively charged DNA wrapped around. Here, these are the histone octamer. This is DNA. Here also you can see histone octamer These are DNA. Negatively charged DNA wrapped around positively charged histone ectamer to form structure called nucleosome. The nucleosomes are seen as beads on strings. See, look at this figure. You can see seen as beads on string structure under electron microscope. Nucleosome forms the repeating unit of a structure in nucleus called chromatin. So this nucleosome, so this is the nucleosome. They forms the repeating unit of a structure in nucleus from chromatin. The chromatin is packaged to form chromatin fibers. So many chromatins together to form chromatin fibers. These further coiled and condensed to metaphase stage to form chromosome. So first nucleosomes together form chromatin. Chromatins join to form chromatin fibers. Chromatin fibers coil to form what? Coil to form chromosomes. Clear? These chromatin fibers coiled and condensed at metaphase stage to form chromosomes. Then there are two types of chromatin, euchromatin and heterochromatin. We'll see what is the difference between these two. In the nucleus, some loosely coiled regions of chromatin. They are light stained. It is called euchromatin. Heterochromatin means in the nucleus, more densely packed regions of chromatin, that is, they are stains dark, are called heterochromatin. So euchromatins are loosely coiled, heterochromatins are densely coiled, densely packed. This is light stained, here it will be dark stained. Clear? Then euchromatin is transcriptionally active than heterochromatin. So this is loosely packed, because of that they are more active than heterochromatin.